Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and we're going to continue our lesson on the cell cycle today, okay? This is actually the last step that we're talking about. The last step of the cell cycle is called cytokinesis, okay? I know that's kind of a long, complicated word, but remember that that prefix cyto means cell, and what we're really doing here is just splitting the cell down the middle, okay? So cytokinesis, up to this point, we've divided the nucleus and we've made sure that each new cell has all the right DNA and organelles it's need. So now all we need to do now is actually separate into two new cells, and that's what this stage is all about. So as a reminder, this is the final stage of the cell cycle. It's called cytokinesis. In this concluding stage, the cytoplasm is divided. So I'm just now realizing that the words cytoplasm and cytokinesis both have that same prefix. That might be a really easy way to remember that. What is cytokinesis? Well, it's splitting the cytoplasm. They kind of have the same prefix there, the same beginning of the word. So that might be an easy way to remember that those kind of go together, okay? So this is kind of a picture of cytokinesis. You can see here that we have two new cells. So these are two newly formed cells. And what I really wanted to point out is just this line kind of right down the middle. You can see that if this is an animal cell, so what we've done is we've kind of pinched all the way down the middle there, and they're going to start to move apart, and that's how we get two new cells through the cell cycle, okay? There's a little bit of a difference in between how animal cells do this and how plant cells do this, and so that's really what we're going to focus on today, is how those two different types of cells complete this final stage called cytokinesis. Okay, so cytokinesis, again, it is the division of the cytoplasm. It will typically begin around the same time as telophase, so this is kind of in conjunction with telophase. It's kind of going on at the same time. This process is going to result in two new identical cells, okay? If these cells aren't perfectly identical to each other, then that means that there's been some sort of mutation happening throughout this process. And sometimes that could be a bad thing. Sometimes it can be a good thing. Most of the time a mutation doesn't really cause anything, but we want these to be as identical as possible, okay? Another kind of vocabulary term I want us to know is this term right here, daughter cells. Okay, so like we're starting with one cell, it divides into two, and we call those two new cells daughter cells because they're kind of the offspring of that cell that we started with. So don't let that confuse you. Daughter cells just means that they kind of came from an older cell, okay? So with cytoplasm, it's the same number of chromosomes as the original parent cell. So however many chromosomes that parent cell had, again, you can kind of see parent cell is what we're starting with, and then daughter cell is what we're ending with. That kind of makes sense, right? Daughters come from their parents, okay? So our whole goal here, all through this process, we've taken a lot of care to make sure that the DNA that we're replicating is exactly the same. So now those two new sets of DNA have gone to the daughter cells, and our last job is just to split it down the middle so that we're ending with two new identical cells, okay? Let's look at cytokinesis in animal cells, okay? So with animal cells, they undergo a process called cleavage furrow. So the cell membrane pinches inward at a cleavage furrow and it forms two new cells. So if you kind of look at that picture in the bottom right, you'll kind of see this process in a little bit of detail. Starting right here, you can kind of see that this kind of looks like two cells, but they're still connected in the middle. So what we start to do is when that pinch kind of starts to come in, you can kind of see it pinching right there. That's called the cleavage furrow. To cleave means to split. So we're trying to split it down the middle. Think of like a cleaver as like the type of knife that a butcher uses to cut the meat down the middle, okay? So that cleavage furrow continues, the pinch kind of becomes more exaggerated, and then the end, when the cleavage furrow goes all the way down the middle, we are ending with two new cells here. Okay, so this is how animal cells do this. And again, not only do these daughter cells have the exact same type of DNA and a identical replica of the DNA, each of these new cells are going to get half of the organelles that we started with here in the parent cell. Remember that in the G2 phase of interphase, when we're preparing for mitosis, part of that preparation is to make extra organelles. So at this point, these two new cells are fully formed and fully functioning. They can do everything they need to in your body. And so now these cells aren't done with the cell cycle, they actually just start the whole thing back over again. Now that we formed two new cells, it's going to go back to interphase and this whole cell cycle starts back over. Okay? So that's how cytokinesis works in animal cells. I wanted to take a second and talk about how cytokinesis works in plant cells. It's a little bit different. So in plant cells, instead of that cleavage furrow, we have something else forming, and it's called a cell plate. A cell plate forms along the center of the cell, okay? The cell plate gradually transforms into a new cell membrane, and the new cell wall develops around the cell membrane. So let's kind of compare this one to the cleavage furrow. This is our cell that is undergoing cytokinesis. Instead of pinching down the middle, that wouldn't really work because this cell wall is really, really tough. So instead of pinching it down the middle, we get something called a cell wall forming. This cell wall is going to continue to form and elongate until it totally splits the cell down the middle, and now we're ending with two new cells. And just like in cytokinesis with animal cells, these cells should be exactly identical in both regards to the DNA that's in them, as well as having their own set of organelles. So these two cells are exactly identical in every way imaginable. That's our ultimate goal. If that's not the case, if something happens, that might cause issues down the line, and we'll talk about those issues next lesson actually, okay? So y'all, great job. Thank you all so much for following along with me. Y'all did a great job today. That kind of rounds out this whole long process of the cell cycle. Your teacher has some other work for you to practice this stuff on. Thank y'all so much for making it today, and I hope you have a great day.